Uh, so yes, I'm the senior research assistant on the Darwin Tree of Life project at the Natural History Museum. Um, as Tom said, I'm a marine biologist really, but over the course of the last 4 years, I've been introduced to a lot more species, particularly in where most of the diversity is, but also delve into freshwater molluscs when we get the opportunity. So what is the Darwin Tree of Life project? Um, Sorry, having trouble changing slides. Yes, there we go. It's really part of the larger Earth Biogenome project, which aims to sequence, catalogue, and characterize the genomes of all the Earth's eukaryotic biodiversity over a period of 10 years. It's a pretty ambitious goal. Now, the Darwin Tree of Life project was really founded as a pilot project to demonstrate the credibility of ambitious genome project goals, but within a single biogeographic area, the archipelago of Britain and Ireland. The genomes we generate are high quality, they're chromosomally complete, and they're freely available for anyone to use, which is the most important part. So it's made up of a consortium of about 10 institutions. The funding comes from the Wellcome Trust, who fund the Sanger Institute, and the Sanger Institute do all of the genome sequencing. You probably know they do a lot of sequencing, including most of the human genome work and stuff like that, and a lot of COVID sequencing as well. But the responsibility of the species collection lies largely with the botanical gardens of Kew in Edinburgh, who collect all the plants and fungi. The Marine Biological Association, who collects all the marine invertebrates, and also some fish. They help us out with that a bit. And we collaborate with them quite. The Natural History Museum, we're basically responsible for all of the treasure and freshwater animal species, although we do tend to do some marine collecting. So why have this project in Britain and Ireland? Well, the most simple answer is that's where the Sanger Institute is. So the boat is easily accessible, and permissions are much easier. But aside from that, it really is, the British Isles really is a perfect place to have this project. This because the fauna has been so extensively studied over a large time period, and it's one of the best known in the world. And it's got fantastic location records. We also have many academic experts and large networks of knowledgeable and enthusiastic naturalists like many of you here, which we couldn't do the project without really. So we can also cover 43% of the family diversity on Earth, which is a large amount. So next slide. So why do this? Why sequence reference genomes on such a large scale? Well, a reference genome is a representation of and organization of the genome of a species. It's quite similar to type specimens in taxonomy, in that they serve for a standard for subsequent genomic studies. And it makes these subsequent studies easier. For example, say you want to look at the genomic diversity of a species. Once you have the reference genome, sequencing individuals of the same or other populations become easier, same species, and more cost-effective, as these can be aligned to the reference genome instead of starting from scratch. So it's a quicker and easier process. It also means that you can create genomes from historic specimens called ancient DNA. This is where the DNA is really fragmented, so small, tiny bits of DNA. And these are specimens such as dried collection specimens. You get it from shells, from mollusk shells, even anything fixed in buffered formaldehyde. So this allows us to see how species have changed over time in past and future collections. So the reference genome allows you to map these small fragments to the genome and reconstruct it. As you can imagine, this is very useful for us at the museum. as it unlocks our historical collections at a genomic level. They're also useful to understand how life evolved, conservation management, how the biosphere is responding to climate change, and to understand gene function and the main mechanisms of genome evolution. We also hope to develop some more advanced methods along the way to make it cheaper and easier, more and more mechanisms. Right, so main slide. So our priorities. The priority in the first phases is phylogenetic diversity at a family level. At the beginning of the project back in 2020, we estimated that there are around 4,000 families in the UK, but we're all at around 1,000. This is due to them being small, hard to find, or extremely rare. We don't want to take the only specimen seen in the wild for 10 years. But we also have other priorities, such as specific research interests, iconic or important species, and protected species. So researchers can request we prioritize a certain species for their research. So how do we do this? Well, obviously the first stage is the sample acquisition. And this is where my team and I come in, along with other project partners such as the Marine Biological Association and Botanical Gardens. Now, there are three main ways we have of collecting samples. First and most obvious one is to use our museum expertise. Our curators have been busy out in their gardens in the last few years on the way to work, and in their favourite collecting areas around the southeast, handing samples to us. They've also been accompanying us on some of our larger field trips, which are another large source of specimens. For us, our fieldwork is very collaborative, it has to be. Over here on the map, you can see all the organisations, well, not all of them, but most of the organisations we've collaborated in the past, we still do. We still collaborate with all of these groups. That's really good. And we also, well, for example, we usually collaborate with Natural England quite a lot, maybe 2 or 3 times a year, joining them on their bioglitches and their various nature reserves. And it's really useful for us. Really useful. It's very fruitful for us. They bring a lot of people. We bring some people. And we collect a lot of species from all areas. But it's also helpful for them. We contribute to species records in their reserves. Many of them have been sampled for a decade or more, and they haven't really had the breadth of teams there before that we bring. We also get a large number of external from the fireplace. This is really useful too. It's a great way to get any of the more specialist species from external sources. They like to find one species we need and send it to us rather than spending time looking for many. So this heat map here is a combination of the Natural History collection and the Marine Biological Association collections. And this is from about 2020. So this is specimen records. Obviously, you can see that we've been covered a lot of the UK, a few more submissions to come. Northern Ireland we haven't covered, but there's other people doing it in Northern Ireland. So we leave that to them with the difficult transport, things like ethanol, bodies of water. So I think you can probably see some of the curators' houses around here on the southeast. Some of our more prolific collectors, Lepidoptera and Coleoptera there. And you can see our dedicated field trips, like to South Wales, to Yorkshire, to Norfolk, and up in the Highlands of Scotland, South Scotland. Obviously, there's a lot of areas around the southwest, so it's probably mainly Marine Biological Association records, but some of them are ours too. Yeah, we've been all over the place really. So when we collect specimens, we require them, or specimens are donated to us, we require them to be identified to species level or genus if the splits are tricky and they need to be resolved by DNA barcoding. We also require them to be submitted with essential metadata. So this is a perfect example of the data we require. So it's got the species name, the location, grid coordinates, but DTF coordinates good too, date, identifier, and Fletcher name. And those are essentially those we need, like anything like habitat or even smell is very useful to us, species. And of course, none of this would be possible without the DToL sampling team. So we've got Inez, our sampling coordinator, Dominic, our research assistant, and me, and Michael's there as well. So he's one of our Coleoptera curators and a prolific collector for the project. This is us on an MOD beach in South Scotland that we got permission. So it's a very nice beach that only we were there. So it's really nice. Turned out to be a really nice day as well. So I'm going to give you some examples of some of our field work. To the left of you, there's Ben. First two pictures in this one. So Benet's one of our genome observatories, in the Highlands of Scotland. Genome observatories are basically where we want to monitor specific places over a long period of time. So we've been here 3 or 4 times already. This is it on its nicest day. Usually it's very misty, actually it's quite misty in the one below. There's lots of midges. But aside from that, it's really a beautiful place to go to. Nate Scott kindly let us use their filter station here, so we get to get board and lab space for free, even if the space is quite tight. You have Park Rock in Reading in the middle top, near Reading, really nice place that we can easily get to on a day trip. We've got more specialist ones like the lagoons of Isle of Wight below that. There's probably some lagoon specialist species there. And to the right of you, we have Lundy Island trip. We've got very lucky with the weather here. Teamed up with the Marine Biological Association, stayed in tents. So we can basically do these longer field trips anywhere, as long as we're reach a roof over our heads and some electricity for lab purposes. If we have these, we can set up a lab anywhere really. So these are examples of some of our field labs. Over to the left top, there's me in our little caravan with a lab set up in there, so we're there for a few days. Look at that, there's Church on the island. They let us use, very nice. We've got the University of Southampton research vessel. They just stopped on that. Below that, in the middle, there's Ben field station. So we've got a lab to be taken as a bed, in case Gavin needs a rest, in case you give them a break from identifying more madly. And then on the right, there's Park Rock. Now, I don't recommend setting up a lab actually outside in very early days, and it's the first and last time we'll be doing this. We tried to prepare DNA snakes and free specimens. This didn't work. However, identification is very nice little setup. Yeah. As long as you have microscope or anything you need to look at them. So what do we do when we get a specimen? So this is a brief overview of our process using it needs to be assigned a human specimen ID. You can see in the bottom left, there's a species with a tiny little piece of paper which has a barcode on it and a number on it. That's the user number. And that gets photographed and database while still alive. We then anesthetize and euthanize the snails and then dissect it. So the shell will keep as a morphological voucher. Things like ID checks, and those will go into museum collections. And that will be kept along with our virtual photographic voucher on our database. We'll then take a small piece for DNA barcoding, and then dissect and flash freeze lentil-sized parts of the specimen in these small little cryo tubes here. And we have to avoid gut. For our participants and our clinicians. Or if the specimen's very small, we can just flash freeze that. But it's quite hard to get DNA from that. Flash frozen specimens, but yeah, hard to get a small piece of that with DNA barcoding. So take we put the tissue sample in tubes at temperatures of -83s or below, and these frozen tubes will be sent to Sanger on dry ice. So the flash freezing part is very important. Right now, this is the best way of preserving and storing DNA, especially if we're storing it for longer periods of time, weeks or months. As soon as everything comes to it again, starts to degrade almost immediately. So we have 3 ways of flash freezing. We can receive a specimen that moves in. It's fairly small. We can just simply put it into our -80 freezers in the electric facilities that are more for things like insects. However, on field work, we don't have access to these. So use these containers called dry shippers. You can see one being charged in the bottom right. They can't actually see the container because of the gases evaporating liquid nitrogen. So we charge these with liquid nitrogen from this big tank. We have replenished almost weekly at the museum. And the liquid nitrogen gets absorbed by these foam cells. That can keep it inside of a container about -170 to 180 degrees Celsius for around 10 days, but we only really like leaving it 7 days since we keep opening the tubes in. And it's a bit risky. You never know when it's going to suddenly release it. Sure. Since the liquid nitrogen is absorbed in these foam cells and it's not flopping around the place, it makes it safe for transportation. That's why they're really useful, although the space inside is fairly limited, so you can only really put small tubes in, put some large ones in, but you fill it up quite easily. And if these shippers fail with frozen samples inside, and it does it almost overnight, it's fairly costly for us. The thawing process damages DNA quite a bit, so it is a risk. And once the samples are frozen, you have to keep them that way. Probably keeping them lower than -60 or -70 is the best. Anything, any higher temperatures like that risks damaging the DNA. We'll also use dry ice. You can see the dry ice here, the tubes on. We can have this delivered to the field by a specialist company, BioCare, and then they will pick up samples from us when we're done and deliver them on dry ice back to the museum. So that's a much safer way if we can afford it, doing it. So that's really useful. The order of this process kind of depends on what facilities we have. Ones are easy. We factory them, store them at -80 until we're ready to process them for sequencing. So we usually do most of our processing outside of the field season when we have more time. So since insect arthropods maintain their structure and color, we can easily photograph them on dry ice, or still frozen, and do all the DNA barcoding and split it apart. Yeah, the joints between the legs come off really easily, snap off, and the joints between the segments usually come apart quite easily unless it's really large. But for molluscs and annelids or anything really with an exoskeleton, it's best to photograph these live quality. And I don't know if you try to dissect the plug at -80 degrees, but it's very hard, so it's difficult to do without smashing it to pieces. So I like to deal with them live. Added bonus of doing it live is that we can remove the shell from the shell quite easily when they're anesthetized without damaging the shell. So we can keep a really good morphological voucher for things like IV theories. You can still put a whole tiny snail in a tube and flash freeze if you needed to, but taking a bit of DNA barcoding would be quite difficult. But they usually just flash the sample, so the whole sample is fine too. We've actually produced SOPs for most invertebrates here at protocols.io. You just search for DToL. So they're designed for most people. even without prior knowledge of anything. So you can get to the lab, cutting up, dissecting specimens and freezing them quite easily. So they're not too technical. I think the gastropoda one was released with the marine metavivisection because that's one of the first ones we released, and obviously it contains gastropods, even though two of the examples are terrestrial and there's one leader branch there. The toxicity is fairly similar. We'd also take vouchers, even if they're not very good vouchers, but if there's some tissue left, like the slugs, we should keep that in F and they could be used for study. Also useful when technology improves and do more things with ethanol species. So the next step after processing is DNA barcoding. So at the museum, we do all of the DNA barcoding for the project, including plants and fungi. So and other places like Oxford, where they'll send samples to us for barcoding. So we do it all. So DNA barcoding is where we take a standardized DNA, which is good for discerning species. It's usually a 658 base pair strand from the cytochrome oxidase gene, which is the last enzyme in the mitochondrial electron chain, so mitochondrial DNA. But there are other genes too that work better for other organisms. This gene is broadly effective in identifying vertebrates. It's not varied between groups of organisms, as we found, particularly the split between species of the same genus. So for barcoding, I don't do any barcoding. There's Jordan there on the left. He does all of our barcoding for the project and the analysis of it. So we'll basically extract the DNA using a lot of lab methods, and then we'll include primers, which capture either end of this cytochrome gene, and then those will be amplified in a machine, and they'll be put in another machine that sequences its DNA. We used to use something called Sanger sequencing. Now we use Oxford nanotore technology. I think it does work really well for large batches of specimens. And once we've validated them, we bet that they're the same species that was identified, or if it's not the one that's been identified and the same genus, we'll go back to collect them. Often if they could be mistaken, or maybe it's a error in what someone put in the barcoding database. So we want this to be bold and see what that comes up with. This is the barcoded life systems. It's a public database. And we upload all of our sequences onto there too. That way they're available for all. It helps fill in gaps in species. So there's a few species we've selected that aren't on the database, we've added, and it helps strengthen the confidence of other barcodes involved. But it has two uses for us. First, it's used for our species identification before we send the samples off to Sanger. And also Sanger can use this barcode to make sure what we send them is in the tube is the effect species once they've sequenced some of its DNA. Currently, we have submitted barcodes for over 3,000 specimens to the database, and filled in some gaps in species coverage along the way, further benefiting research. Yes, and after that, they get sent to Sanger. So the next step, after processing the specimen down to small frozen tubes, sorry, looked at the wrong bit. Don't know too much about the. It will give you a layman's outline, but I'm a layman myself. So after specimen acquisition, by the way, this is from a sort of the white paper we have on the Darwin Tree of Life project called Sequence Locally, Think Globally. So it's a good one to read if you want more detailed information about the project, and that's where this graphic's from. So after the sample acquisition and data recording, the ID and the barcode validation, we've There's Sanger. From here, they take the tissue sample and they crush it, still frozen, and then they use various lab kits to extract the DNA. They then take an extremely small volume of this DNA and put it through a machine which evaluates the DNA quality to see if the strands are long enough for the purposes of genome sequencing. If they're way too small, then there's no point in proceeding. Yes, then they put the DNA in another machine if they've got long enough strands, and it shears it down to the size they need. I'm not sure why they need this size, but it shears it to around 20,000 base pairs long. So these are much longer than 158 base pairs. And they then make a library, which is essentially a buffer solution with the DNA strands they've extracted inside of it. And then they load this into a machine that reads the sequences over and over again for accuracy. And it tells they are, so C or T, and what order they're in. They end up with strands of DNA in the solution at approximately 20,000 base pairs, called long reads. That's one bit of information they need. The other bit is called HiC, or highly contiguous data. And they need this to identify how the chromosomes are organized, so how all these strands are connected together. They also sequence the RNA, which are the genes that are actually expressed in the organism, so they're more functional. So they can understand the functional DNA, and then they can map this to the genome. When they get all of this data, they put it all together, called assembly. So you can see them using this HiC, the long reads together, and then they can find out which chromosome it lies on as well. And then all of this data is similar to DNA, which is the European Nucleotide Archive, and it's freely available to everyone. So once the data is on there, anyone can use it. They use something called Ensemble, I don't know anything about, but they use it to annotate the gene with its features, so what gene do what, and that's really useful for that. And then the genome is released as a genome note, and the specimen collector, you see here, this is one of my genome notes, my first mollusc, the variegated gallop. So they released these genome notes, and the specimen collector given the first refusal on writing a short introduction to the species, and being first author on the genome note. So I'm first author by writing the introduction and collecting the species. It's a really nice thing to have. These data notes are freely available in the Wellcome Open Research platform. Yes, and that's my first one. That was 2020. So what data have we produced so far? So to date, got over 1,000, almost 1,200 whole genome assemblies. They've been released, not necessarily genome notes, but assemblies that you can get on the ENA platform. We've got over 3,300 barcodes involved. And in the past year, we've generated almost 4,000 sequences. Quite a lot. So biological records wise, we've got detailed specimen collection records with location and everything, totaling over 10,000 specimens in the last 3 or 4 years. They go on for NHM data, they go on for BBIP and laboratory, along with other data. That contribute normal specimen records as well, and plenty of protein tubes at Sanger pending. So I don't really have the numbers for those. You can see a bit more on this. This is the DToL data portal at portal.Darwin Tree of Life.org. And you can track specimens through the pipeline, so if they're ready or pending. So if it's something you're collected, you'll be able to search and see it's Sanger. You wait till it's later until it's made its way to Sanger from us. And then this is European archive. This is where all this data goes. So you can just download a lot of different data there. EBI.act.uk. It might be a bit complicated if you don't know anything about genome sequencing like me. So I haven't been to look at the data yet. And lastly, there's Genomes on a Tree. This is a Sanger website, nothing to do with us, but it's more to do with the larger Earth by Genome project. And this lists everything, or almost everything that's been sequenced on those records for sequence barcoding or genome sequencing, bigger status with all species collected there. So that's a really good resource they have. Find out if there's genomes available, if you need to work in genomes. That's great. .genomehubs.org. So that's where all the data goes, really. So by now, I'm sure it's desperate for some mollusk content. There hasn't been much. So here's a lot of plants. These are some of the best photos we've had from our processing over the last few years. These are ones we've collected. So there's a range there, but there's many more that we have done. Let's have a look at some figures. How many have we really collected? So these figures are from UKSI and may contain errors, but it's an idea of where we're at. If there's any discrepancy in the numbers, I apologize. It's changing all the time, but that's about right. So according to USI, that's 2,058 mollusk species in the UK. A large proportion of these are marine, with freshwater or brackish at 105 and spectral at 170. So this is how we're doing. So admittedly, we're not doing as well as we'd like, having concentrated a lot of our field work on and not engaged as much in collecting mollusks, but we hope to do that. Here are numbers so far. So we've got 151 marine, a very low percentage of the total, but it is harder and more costly to reach the subtidal species. We don't have much budget, hire boats or anything. That's probably why most of them have probably been intertidal species and common subtidal species collected by the Marine Biological Association. So these aren't just our numbers, they're project numbers. We've got a fairly decent number of freshwater and terrestrial, although I'm sure we could easily be doing better here. And we'll push for more this year. So our diversity figures is how we're doing. We're doing okay diversity-wise, but our family coverage is a little low, under a third, and that was one of our priorities. So we definitely need to get out there and collect some more mollusks this year and the next year after that. So this is a bit of a call for help. We need your help. Hope to engage a lot more with the Conchological Society this year. So if you'd like to collect us, please let us know. You can express your interest by contacting us at Darwin Tree of Life, nhm.app.uk. This is a good email address to use that goes to all of our sampling teams. So if someone's away, they'll pick up your email. We can send you lists of what we need. Tom kindly looked over our freshwater and freshwater list. Made that good for us. So send that to you, and we require up to 5 individuals each species. If something goes wrong, or if it's very small, they probably can't get the amount of DNA they need, the genetic material they need out of one specimen. To find live specimens, you've identified the species or genus level, and you'd like to send them to us. Send your form fill in, so you can put in all the metadata. And then the animals need to go in a ventilated container, so it's on its favorite habitat. External labels may be required for working snails. The warning, we received one and the snail had eaten the label. There's only a tiny bit left. Luckily, it was an easy to identify. So let us know when you post these, and avoid posting close to the weekend. We won't be here to collect it, or during peak days. Very hot and they unlikely to survive that. So finally, I just want to show you a few interesting specimens I've collected for the project. So back in 2021, the University of Southampton finally gave me a cabin in one of their research seasons, the Porcupine Bristle Plain, which sit there off the southwest of Ireland. Technically, it's international waters, not within an island, but we're including it in the project anyway. It's a very well-studied area. They go back annually, and when they can, they do see falls. So, and it's up to 4,800 meters deep, and it's plain. It's very it's 4,800 across a lot of it. Yeah. So this is Grimpetus discovery. It was only described in 2003. We've got a couple of these. Obviously, they look a lot nicer when they're swimming around. Unfortunately, for all these tools, most of the things are already dead, so the DNA has degraded a bit. But it's more of a R&D thing, development thing at the moment. But hopefully we can get at least something. We've got at least the barcode from it, so we added that to the barcode database. And I think it's the only one for that species. There's one for the genus. That's really cool things to find. Other things from the trip, so Stuffy Fetus Viticola, so that was some really nice specimen with some of the arm for sequencing and barcoding. So they're at Sanger now. There's another one, Euphonium megalops, really nice find. So I thought I'd show you some nice pictures I took. So this is in the UK. This is now my favourite snail. I've never seen one with a tiny little shell on its back. That was amazing. Fence bust from North Wales, or the Bardia Ruta. I've got to show you that my favourite snail now. And I think hard to find. And this is the favourite thing I've collected. This is Niederbank, Oceania Rosa. This we found on the Lundy Island that I've snorkeled down to about between 5 and 10 meters. I saw this thing motoring across the fan, so I grabbed it and brought it up. It turned out to be this. I was extra excited because in the UK, the record, I don't know how old the record is, or if anyone's found one larger, but the record is 60 millimeters, so this is 100 millimeters. Although in Norway, it's been known to get 20. So that was my favourite one, and that's then the genome sequence. And this isn't DToL at all, but this is just a trip that I went on. I thought I'd show you. It's really cool. So DFAS have this project, DToL project. They kind of gave us a couple of cabins on, and it's really to monitor and protect the UK overseas territory. Currently, UK overseas territories aren't within our remit, but we're hoping they will be. So we went out there to the Central Island and St. Helena. We found this particular species called Berta, now in our collections. Sampled a bit of DNA and genome sequencing too. So hopefully that will be good. But one of our fish curators got the idea of someone else bring a using pen along to shine at the photophores around it, see what happened. And we got this useful red glow. Once we turned all the lights off, I just took a long exposure, shine the torch all over it, and these really nice pictures. Interestingly, most photophores are shine blue when they're live and underwater, but for some reason, most of them only shine a UV pen on it. They turn out red, but not quite sure what they might be structural. Yeah, I've got to show you that. I'd like to thank all of our project partners and all of the people that supply us with identified specimens. Again, that's not all of the people who've worked with us, but most of them, so apologies if I missed any groups. Thank you to the Welcome Trust for funding it for the last 4 years. Really great. Thank you for listening. So any questions?
Thank you very much. Thank you, Chris, very much indeed for that fascinating talk. Certainly, there's a lot there that I had no idea of, and I'm going to take turn to and ask you a couple of questions. But I'm a little bit unclear about the benefits of the DNA sequencing and the genome sequencing. One seems fairly straightforward, one highly complicated. what more is going to be, the benefits?
Well, that's a good question. I can answer a bit of it, but obviously there's a lot more going on in an mitochondrial DNA. On the whole genome, so you can find areas of the DNA that are used for set purposes, like sensory things, in snails, stuff like that, and you can actually trace back to where these genes evolved and where they came from. If you manage to get some DNA out of ancient specimens, you can trace, or indeed the DNA from lots of different closely related groups. Find out where these genes diverged from. Also very useful for the genomes, very useful so you can reconstruct. So you can get ancient DNA, you can crush up cells, and they contain DNA from the organism itself and from the environment around it. And if we have genome sequences, whole genome sequences for these organisms, we can go back and see get a glimpse into what life is like there and how they've adapted since. You can do things like look at how the environment's affected its genes. So you can see the stress that things like climate change and pollution are putting on them, and you can see how they adapt and respond to that. It's also useful for conservation, so you can find the species that are quite fragile and can't adapt as well as other organisms, and you can adapt conservation strategies to that. So that's just some of the uses, but there's a lot more, but I'm not too involved in them.
DNA sequencing is for identification of the genome. It opens up the field in a unlimited way.
Yeah, it helps with the DNA barcoding is quickly easier. Yeah, and it's all
The other question I was going to ask, you showed a picture of the ramshorn snail of Panelvarius ornus, which is one of the red varieties. Now, this company, do you do repeat on different specimens of the same species, or once you've done a species, really that's it?
Yeah, so we can take research requests. They will have requests for two different populations, but it's handy people. Yeah. So actually, the two different parties will say yeah.
So that ramshorn, for instance, you might do the red one and the black one, and see whether
whether they might do the whole genome from scratch one, and the other one is so scaffold is together a bit more easily for the other one. Yeah, we can request things about it.
So let me throw questions open to people present here or online. Anybody got any questions?
Yes, can I ask a question? Richard.
That was, how do you check on hybrids?
Good question. I'm not really sure, because they obviously we come across this with fish, hybridization tracking fish. I'm not really sure what strategy they have for it. Right now, we're kind of focusing on the less tricky things to do. So yeah, I'm sorry I can't answer that, but I can find an answer for you and email it. Yeah.
Can I ask a question?
Yes.
Can I ask about type localities? I'm interested in your sampling strategy. You seem to be just going out and collecting things willy-nilly. Do you think it would be sensible to try and get your sequences from type localities so you can directly link the taxa to their type locality, for example? Because we're rather assuming that people like us can tell you exactly what the things are. We might not always be able to tell you what you might tell us about the genetics. There might be a mismatch between what you get and what we think they might be. So it's quite interesting to go to the type locality to see what the species is there.
That would be the golden example of what it's like. So it would be great if we could go and get specimens from the type area localities. To my knowledge, we've only done that once, but I'm not sure what other people have sent in. We went to Isle of Wight and collected the I don't know, the starlet sea anemone, and that was, even though that's probably from America, it was first described in the Isle of Wight. So we did go on a specking trip to do that. I think the main reason we don't is just ease, really. It's a lot easier to collect this huge volume of specimens in a fairly short amount of time just by going out and collecting and seeing what we find. So yeah, it would be great if you could get everything to be type localities, but it's not always
But
feasible.
there's not so many type localities in Britain. I mean, most of them for the non-marines will be Sweden, presumably, because Linnaeus described most
Yeah,
of the things from Uppsala. But just where we can, would it not be interesting to focus on that? I mean, there are a few I can think of have their type localities in Cambridgeshire, for example,
yeah,
Cernuella virgata, Pysidium hensloanum, Pysidium nitidum, all those described by British people. It'd be quite straightforward to collect
yeah.
those from the type
Okay.
localities, and I think that might be a preferred sampling strategy.
Yeah, that would be great actually. Yeah, if we get list together type localities and they're not too far away, or people can help get them and send them in, then that'd be perfect. Yeah, I do agree. Yeah.
Doesn't always work. Peter Dance and I went to look for the original description in Britain of I think it was Porzilia bidentata, and in Wiltshire somewhere, and absolutely no sign of anything around in that area originally described. Right, more questions. Peter.
Question about you anesthetize the molluscs, and then so what happens when you anesthetize them? Is it a bit like killing them, the muscles relax and then you can release them?
Yeah, so well, but things like snails, it's great. Once they're relaxed, we can remove them from the shells, and then usually I just dissect its head and then put that in ethanol and use the rest of the specimen um, to for the sequencing and everything. It's a bit more difficult with the smaller things. They can anesthetize, and then you can take a small bit and then freeze them. We can also actually just once it's out of the shell, just flash freeze it. If we've got the dry shipper out, and if it's large, we need the dry shipper really to freeze it properly. So that's the best way of doing it. You can use a variety of different ways. It's not ideal. I prefer to just pressure freeze them, but it makes the process quite difficult.
Yes, hello.
You had a question.
I was going to ask something similar. What exactly do you use to anesthetize a snail without
You can
damaging
use,
the DNA?
yeah, so you can use a small sensitive ethanol solution, so like 5% ethanol to 10% maybe sometimes.
Okay.
Take a bit of time. You could also put the terrestrial ones in water, and then you can sprinkle some not menthol crystals on top, and that will eventually anesthetize them. So it takes a while.
Yeah.
There's also something I've forgotten the name of it that we use. The Marine Biological Association use it much. They got a healthy supply of it. Something beginning with P, and that works quite well. And also use magnesium chloride as well. But different things work better for different organisms, and there's usually a bit of time involved. But they do. When I first started doing it, I thought they had died, but then when I take them out of the solution or rinse them off in water and put them back, they eventually wake up. So, so I was glad of that. So yeah, there's a few ways of doing it.
Okay, thanks. I did have another question as well, if that's okay. So yeah, you ask for 5 individuals of each species.
Yeah.
Which can be a tall order for even some quite common things that take some time to find.
Yeah.
But you've obviously got some good results where you've just found a one-off specimen. I'm guessing you didn't have 5 of each of those types of squid.
Yeah, no, a lot of the things we have one specimen for, and a lot of the time they can work. If they don't work, they always provide us with a list of specimens they need resampling. So you can always just collect one, and if it needs resampling later, they can do that. Thing is about the genome sequencing, it's all been done from like insects on animals, humans and insects basically. So it works very well for the insects, which is why a lot of our Lepidoptera numbers are quite high. But molluscs is a bit of research and development, getting the right techniques for it at the moment. So hopefully it should get easier, and they'll just be able to do the really small ones just with one specimen. So it's only really 5 if you can. If you find one, that's perfectly fine as well.
Well, I think we ought to praise Chris and the whole project for such an ambitious program. I think it's beyond the bounds of understanding for most of us quite how enormous it could be. It's great that he is involving the Conservation Society, and I would like to thank him for including our logo of the various things he's shown us. It might be useful if you could let us have a list of molluscs that we still want to have, an updated one, because it will have changed since the original one. And I only had the terrestrial one, and I'll make sure you get the email contact of our marine recorder, because Simon would almost certainly benefit from having the list. And then we can encourage people perhaps to ask, or we could do an article to our next meeting. Yeah. It would bring it to much wider notice than those people who are present today. That'd be brilliant. So that we can try and get more people to participate in this wonderful project.
Well, thank you. We do need everyone's help. So yeah, very appreciated.
So thank you very much. I think we'd all like to thank Chris in our usual way.
Thanks for having me.